Okay, so in this third segment, we are gonna talk about another mechanism in which antibodies can be used to carry out their effector functions and clearing of pathogen, particularly bacterial pathogens. I want to point out that this is sort of a crossover between innate and adaptive immunity because I'm going to introduce a part of complement that doesn't need antibodies and is part of is a part of um, really your innate immune response to that's able to fight bacterial infections. Um, so this area, this type of immunity is immune mechanism is referred to as complement. It's referred to that because it complements the other types of immunity that you have. It's a complementary process. It's probably pretty ancient, as we'll see, and provides a kind of a different kind of immune reaction that can occur, and it's quite important because it has a very powerful it's a very powerful in terms of its antibacterial function, and we'll see later it's also important because of its uh, a type of hypersensitivity. Hypersensitivity type 3 reactions are also dependent on complement. So we're going to come back to that. So you need to know what complement it is to understand its antibacterial function, how antibodies are important to it, and then as we'll see later in hypersensitivity. Now, complement, there are three major biological activities associated with complement. There's the activation of phagocytes, including macrophages and neutrophils. There's the lysis of target cells, and there is the opsonization, that is the deposition of antibodies and what are referred to as complement proteins. One of those is C3b, or microorganisms and immune complexes for recognition by cells with complement receptors. Okay, so those are the three sort of processes. We're going to lay down proteins on cells that are going to cause a eventually a destruction of the cell. We're going to use antibodies. We're going to see how antibodies can direct that, and we're going to see how phagocytes can be a part of that as well. So it's a pretty complex process, and so don't I as we go through this, remain calm. Don't worry too much about uh, don't worry too much about it. Just pay close attention to the things. I want you to pay attention to the things that are similar and different between the two complement pathways. But if you go through this, and as we'll go through it, you'll see it's actually can be organized, even though it's complex, into discrete areas that you can get your hands around. So there's 20 proteins or more that are involved. Some of those are enzymes, some of those are components, and some are proenzymes. The components are part of this complex forms a site on the protein that in the end cause dissolution of the cell by putting holes basically in the membrane. There are two pathways, complement pathways. The first is referred to as referred to as the classical pathway, and it's initiated by a bound antibody or antibodies. The second is referred to as the alternate pathway, and that does not need antibodies. It's initiated by bacterial cell surface components. And both processes, important to understand, and undergo an amplification phase but converge on the same final pathway, final step. And so it's sort of like thrombosis in the sense that there is initiating small steps that happen initially, and then there's an amplification in which you get an amplification of the reaction and an increase of the reaction, great increase in the reaction as time goes by. In the end, both processes um, uh, generate what's referred to as a macromolecular membrane attack complex, referred to in the shorthand as a MAC, and that MAC is responsible for the lysis of a variety of cells, bacteria or viruses that may have developed this may get this into their surface. So if we look at the classical pathway first, if we have a bacteria or tissue and we have antigen on it, antigen that's on there, um, you can bind, those can bind potentially to IgM or IgG. All right, and if they bind to IgG, they will bind and let's see, get this. They will bind, and their FC regions here, right? FC regions are going to be accessible. They're going to be sort of pointing outside of the. They're going to be pointing outside of the where it's bound. For an IgM, it's if you like, it's like your hand. If you took your hand and you tried to put all your, if you just do this, take your hand and put your fingers on a table, what you'll have is you'll have uh, what will be unveiled or made present is the FC regions, the combined FC regions of the IgM. And it's almost like a platform, right? It's almost like a little table. And that's going to be important, as we'll see. Because what binds to that is a molecule called C1q RS R2S2. And the C1q binds to the molecule. It's a big molecule that we'll see that is a component of the is the first component of the complement called, that's why it's called C1, and it is in the blood, and when it sees the surface of an IgM that exposed IgM bound to a tissue, a bacteria tissue, it's going to bind to it. If it sees two antibodies that are bound to it, and they're the same and they're within the length of the protein, are able to bind it, it will do that as well. Now, in terms of activation, it turns out of complement that IgMs, because you notice here they're the best at activating complement, because they have this very large surface that can be recognized by the C1q. For IgGs, for example, that are bound to a surface, the antigens have to be placed right. So in order for you to get the right in order for the IgG FC regions to be in the right distance apart, right distance in order for complement to bind to it. So what this means is that, let me just go back here a second. What this means is that it takes 1,000, in general, it's about 1,000 IgGs bound to, say, a bacteria to equal the binding of one IgM, because in terms of the probability that you're going to have the right dimensions here, okay, in order for C1q to bind. So if you have IgM is big and it can bind across in several different distances because it has all these binding sites, and its FC region is potently binds to C1q. There's a much higher probability that it will bind because it has all these binding sites, as opposed and have the right and expose the platform for C1q, as it is for IgG to be in the right have the right distance for it to bind. So in general, IgMs are the most potent complement activating antibodies. They're more potent than IgG. And you'll find that in the early days of an infection where you don't have much IgG specific antigen is going to be produced, the IgM is really important because that's going to be the one that's going to carry out the immune function, and you're going to see here the complement activation. Now there's another protein here, this R2S2, this little ring, this donut, which is assembles around the C1q, and that is when it does that, it forms an enzyme. And so that an enzymatic activity, it's able to carry out enzymatic activity. And so it's this whole this whole complex that's important. The C1Q, which is this J shape, these two J-shaped structures, and then the R2 S2. So this comprises the whole the whole complex that's needed to carry on the next reaction. So going forward, then when those are bound, they will now, because they have enzymatic activity, they carry out an important function, and that is the hydrolysis of two other components that are in the bloodstream. And I might add that most of the complement components are produced by the liver. And those are C2 and C4, complement protein 2, complement protein 4. And it cleaves them into two fragments, A and B, 2A, 2B, 4A, and 4B. And so right away you can start seeing you've got an enzyme, and enzymes can carry out multiple turnovers, and so you can start to see you're going to get more, you're going to get more, for example, C2A and C2B is going to start being produced by C1Q. And so this is one step in which you start to see amplification, so enzymatic amplification of that the reaction. This function is now referred to as the C2-C4 convertase. So when the C1Q R2S2 is bound and is doing this, it's now called the C2-C4 convertase. Now, the C2A and C4B are going to bind to the surface of the cell, the bacterium, and they form a complex, and that's what this is referred to. They form a complex referred to as C2A, C4B, and that complex on the surface of the cell is referred to as C3 convertase, because it's now going to carry out hydrolysis of the C3, it's essentially a proenzyme, C3 to C3A and C3B. And this turns out to be one of the critical factors. And so if you're going to get lots, so you're going to have, say, one of these, you're going to have now lots more of the C2A, C4B than you had the C1Q. Then you're going to get more C3 is going to start being turned over, so it gets further amplified. So you get this another enzymatic amplification of the reaction. And the C3B is important because it now will combine with the C2A and C4B to form the C2A, C4B, C3B, and this forms another convertase. And that convertase is, well, there's two things. That convertase, let's see if I get this in the thing here, is C5 convertase. And so this C5 this will carry out the hydrolysis of C5 into C5A and C5B. So C2 to C2A and C3 to C3B and C5 to C5A and C5B. Now, one other feature of this is that the C3A here and the C5A here will go and activate mast cells. And mast cells are important because they will initiate potentially histamine release. And they also activate further an immune reaction in terms of immune response, in terms of activation, as well, as other cytokines. And I just want to point this out. We're going to come back to that when we look at type 3 hypersensitivity, and we'll talk about mast cells, and we'll talk about histamine release. I want to introduce the concept here, though, that these other factors are important because they activate further the their immune response. And so what ends up happening is on the bacterial cell or tissue cell, um, this uh, uh, your C5 convertase, then you start laying down C5b. And C5b turns out to be a structural protein. And so the C5b will now adhere to the bacterial cell. And when it does that, it has basically a half-life of about 2 minutes or more, about 2 minutes. And if it doesn't see C6, it will come off. It's sort of a fail-safe way to think about it. If there's not enough C6, then you don't go further with this, and this may occur on the tissue cell, or you may have that destabilized on the tissue cell. But on bacterial cells, it will adhere. And so what ends up happening is this protein C5b will bind to C6, C7, and C8, and they'll form this kind of dumb shell. Will combine with these to form this kind of dumbbell-shaped protein that then allows for C9 to start to bind to the surface, to come into bind to the surface of the cell, and it basically forms a nanopore around that set of scaffolding proteins, C5a, C5b, C6, C7, and C8. And that's the point. That's the point where now you have a pore has been made, and this structure is referred to as the MAC complex, macromolecular attack complex, and this initiates in the cell a lack of its ability to control its membrane and what things go in and out of, particularly water and other nutrients. And so the cell becomes unable to control that, and as a consequence will die. And so you ended up getting cell lysis. So in this case, what you see here, you can see here's a membrane of a cell, and you can see all these little MAC complexes that have been formed on the surface, and they're all they're basically little nanopores about 10 nanometers apart and about 15 nanometers high, and that will allow fluids to come in and out of them. And you can see here, this is a membrane lesion where you can see the side, and you can see there's a tube that goes right through this membrane. And so that is referred to as the MAC complex, and it is one of the important, it is the critical kind of way that you see that it's the critical thing that complement does in terms of destruction of the cell. So if we look at this in total, right, if we put this all together, we see binding of the antibodies to the antigen. This is where antibodies are important, right? This is the binding. This is where antibodies do their, affect the response. Binding of C1q, conversion of C2 and C4 to factors to C3a and C3b that binds to the surface. That's what this little bar means, binds to the surface of the bacterium. The conversion then of C3 to C3b, further converting this protein complex into a C5 convertase, conversion, and most importantly C5b. And in C5b, it's a oligomerization with on the surface, it's a oligomerization with C6, C7, and C8 to form this scaffolding set of proteins that allows then the assembly of the MAC complex in the pores and then cell lysis. So you can see it's a pretty straightforward process. And also I just want to point out these factors will go on and are immunostimulatory and stimulate the mast stem cell and stimulate other cells in the immune response, particularly mast cells. Now, there's the alternate pathway. So I just want to point for the for the alternate pathway, there's going to be things that are very, very similar, and then there are going to be things that are a bit different. What's really similar is the MAC complex formation, and then sort of the formation of that to form that. So the end product is the same. There are some differences in how you get there. And so let's take a look at um, the alternate pathway. Now on bacteria, bacteria, um, uh, bacteria don't have bacteria. It turns out that C3 can undergo spontaneous hydrolysis. There's actually a thioester that holds this protein together, holds the protein, these two subunits together, and it can undergo spontaneous hydrolysis. If it does that, if it's doing that in close to a bacterial cell, it can actually adhere to that cell, bacterial cell. It does not do this to mammalian cells because mammalian cells have sialic acid, which creates a more of an acidic environment. Sialic acid is a sugar that it usually terminates a lot of oligoglycans on the surface of mammalian cells. And when it does that, it creates an environment that's fairly acidic, and C3b is quickly removed from the surface. However, bacteria don't produce that glycan, and so they become more prone to having C3b adhere to their surface. So, and here you have very low levels. Bacteria have little or none yeast, and that environment is is a way of preventing binding of C3b to the surface uh, of mammalian cells. Now, C3a, of course, can go, and if this is happening, you can get mast cell or immune cell activation. Once that's formed, a protein C3b, if it binds to that surface, can form a protein called B, which has two subunits, BA and BB, can adhere to the surface as well, this B factor. And there's a protein called D that can cleave that B into BA and BB. And when it's on the surface, it's activated to do that. And BA can also go and do some immune cell activation like mast cells. This will form a complex called BB and C3b on the surface. Now, another protein called properdin, Properdin, properdin will bind to that, is sort of catalyzed to bind and assemble on the surface, and it's a structural protein to stabilize that interaction between the BB and the C3b. And so in that process, what you form now is a C3 convertase, and this enzyme that's on the surface will start converting C3 to C3b and C3a. And you can see here that it's a pretty rapid reaction. There can be over 2 million C3b molecules are produced on the surface in 5 minutes, so it can be really quick. And this is where, again, this is amplifying. You get several, one enzyme can produce thousands, millions of those molecules once it's on the surface and activated in minutes, because it is a high catalytic activity. So this is referred to as the C3 convertase. So you can compare this C3 convertase to the latter one. Notice that C3b is common to both of them. Just a couple of the other factors are not. Now, the C3b will, you increase the amount that complex can, that complex will form, you can add additional C3b to that complex, and when that happens, that complex will now carry out C5 convertase activity and convert C5 to C3B, C5B, and C5A. And again, the C5A can carry out immune cell activation. With the C5B now adhere, ready to adhere, produced can adhere to the cells. Now, pretty much everything is the same as we saw before. So there's a bit difference here. There's just a difference in the B protein and the proprotein, and a little bit of difference in how those are produced. So the common thing between the two is going to be the C5 between the alternate and the classical, and the alternate is going to be the C5B forward, and the thing that's different is how you get to the C3 convertase, okay? And the fact that the alternate pathway does not involve antibodies. And so you get C5B now on the surface, and now that can again go through what we've already discussed, in which you can self-assemble C6, C7, and C8 to form the scaffolding protein, which will then lead to the assembly of C9 to form the MAC complex and cell lysis. So you've seen this all before. This is pretty much the same. So this is the same. So when you're looking at these two mechanisms, what you want to do is look at what's the same, and then just map out what the differences are. And the differences are in just the error in the initial activation getting to C3 convertase. So here's this one sketched out, and you can see here again that if we look at this, the things that are the same between them, between C5, the classical and the alternate pathway, or those things. The things that are different are how you get to the C3 convertase, right? These things, right? So those things that are different. And in the alternate pathway, it is truly part of the innate immune response because there's no antibodies that are involved here. There's no adaptive immune response. And so this is an intersection between, as you can see, between the adaptive immune response and the alternate immune response. And the differences in the proteins between the proteins are, in the classical pathway you have C1q, RS, R2, S2. That's different. And you have C2 and C4. That's different. For the alternate pathway, you have the B factor, proprotein, and the D protein, and that's what's different. Those are the two different. So just pay attention to those differences. So one other thing is that phagocytes also contain C3b receptors on their surface. And this is important because this means then that if you have C3b on the surface of a bacteria, if it starts laying it down, C3b is on that surface. Complement is working to destroy the alternate pathways, working to destroy that the bacteria. But a phagocyte will recognize C3b on there and will also potentially engulf the bacterium. It marks it as something it needs to destroy. So we're going to stop here. You want to stop? You can stop the video. Here's some review, 3 short review questions you can take a look at and answer, and see if you know it. See if you know the answers, and can go back and review. In the next slide will be the answers. So if you want to stop here, that's good, and then the next slide will have the answers. And we are now at the end of the third part, third segment.